Hi there guys, and welcome to another A-level biology video. This is Mr Phillips, and I'm doing video, I believe 2.1 this today, which is about cells, animal plant cells and their ultrastructure, which is the organelles in the cell. So really quickly then, that's all we need to know what an organelle is. Well, if you'd have done organisation at GCSE, you would have been told that a cell makes up a group of tissues, a group of cells make up a tissue, sorry, a group of tissues make up an organ, and organs make up an organ system, and they make up an organism. Well, below the cell level are organelles, and organelles are part of the cell that have a specialised role in playing, a specialised function in playing an important role in the parts of. So if we start off then, we're going to look at animal cells, then plant cells, and look at the function, summarize the function of them together. So you might get a picture which looks like either of these. So you might get a sort of a cartoon diagram, or you might get an actual electron micrograph of it. If we go through the cartoon diagram as much as we can, and then we'll go across the electron micrograph to sort of recognize it. So if we have a look at the diagram, obviously same as GCSE, this is from an electron microscope, so it's called an ultrastructure. So we think it's the ultimate thing we won't discover any more. You've got the nucleus. Okay, now the nucleus is obviously not just a little ball in a cell. We've got a bit more detail. So over here we have the nuclear envelope. Okay, so the nuclear envelope controls what goes in and out of the cell. So the nuclear envelope would be this bit here on the animal cell of this micrograph, okay, and it controls what enters and leaves the nucleus. So you'll hopefully know soon that DNA never leaves the nucleus, but messenger RNA does to make proteins. Now within the nucleus, there's darker patches called the nucleolus. Now we haven't really got a clear nucleolus on this animal cell here, but it might be starting to sort of form here. A bit of a darker area. And what we haven't got labelled, but I think is quite important, is this area here of the cell. So I'm trying to find my pointer to point it out to you, and that is the nucleoplasm, which is like the cytoplasm of the nucleus. Okay, so that's that. Then in the cell, obviously we've got the cytoplasm, which is all of like the chemicals, where the enzymes occur, where the chemical reactions occur. Can't really label that. But then we've got the mitochondrion, or individually mitochondria, or the other way around, I think it's me. And your mitochondria are these structures here. Okay, so relatively big structures in the cell compared to other parts. Obviously, nothing compared to the nucleus. But you've got lots of mitochondria in this cell present. Obviously, mitochondria do aerobic respiration to produce ATP for our energy needs. We then have the smooth endoplasmic reticulum, which smooth endoplasmic reticulum are involved in lipid production. So the production of fats and also the production of vesicles, which you'll learn about later on in the course. A lot of people forget the smooth endoplasmic production. It's difficult to see on our diagram. It all looks like me that they're rough endoplasmic reticulum, so we'll label that next, I think. But you would have to be definitely smooth. You'd have to see it quite easily. But over here we've got rough endoplasmic reticulum. So rough endoplasmic reticulum is a lot of membranes with ribosomes attached to them. Okay, and that's what you can sort of see all along here. Okay, so rough endoplasmic reticulum. They're involved in protein synthesis. On top of that, we've got something called Golgi apparatus. So the Golgi apparatus, which we'll talk about later on in a bit more detail, basically they will, when we make proteins, we want to send them out of the cell, they will package and they'll modify proteins so they don't get broken down in the bloodstream or don't get damage leaving the cell. Okay, then we've got some extra things. We've got something called a lysosome. Okay, so a lysosome which contains hydrolytic enzymes, and they're the cell's defense. If you've got white blood cells, they'll have big lysosomes to try and destroy any bacteria when they do phagocytosis. And then we've got the cell membrane. Obviously, the cell membrane, hopefully you know, in an animal cell, controls what enters and leaves the cell. And then the very small dots up by the rough endoplasmic reticulum and attached to rough endoplasmic reticulum are ribosomes, which are involved in protein synthesis. So if we look at plant cells, obviously we've got a lot of similar structures in plant cells. So Golgi apparatus, so I'm only mentioning these briefly again. Golgi apparatus also occur in animal cells and plant cells. They are involved in repackaging and modifying proteins. We've said about ribosomes, plants need proteins as well. So ribosomes are involved in the making of proteins. That's why proteins are synthesized or made. The nucleus is made up of the nucleolus, the darker bit, and it's made up of the nuclear envelope and the nucleus. The clear nucleolus on this cell, okay, is this bit here. So that's your nucleolus, okay. So we will label that as the nucleus. It isn't. The nucleus starts where I'm drawing now. So this is the nuclear envelope. This whole thing is the nucleus, and then the nucleolus is the dark bit in the center. Okay, we've got rough endoplasmic reticulum, which are again just have protein ribosomes on them to make proteins. We've got a cell membrane, and I'll just label the cell membrane in red here. So the cell membrane here now on this micrograph, it's quite useful, I think, because you can see specifically the cell membrane, which is this line that I'm going along here, is the cell membrane. And then obviously you've got the cell wall. So the cell wall, which I'm labeling in this green color here, you can see is actually a very faint line there, which is the inner bit of the cell wall going along there and obviously joining up alongside the membrane, and there's the other line of the cell wall there. So your cell wall is lying on the outside of the cell membrane. The cell wall gives a plant cell structural strength to make sure it can stay turgid when it fills up with water and it doesn't burst. We've then got a vacuole. Okay, so the vacuole holds glucose and water, or sucrose sometimes, and water. So it's what we call cell sap. And on this second cell on this diagram, you've got a vacuole here, and the vacuole's membrane is called a tonoplast. Okay, so T-O-N-O and then plast, tonoplast, and that controls what enters and leaves the vacuole. Okay, we've got then starch grains, which are present obviously because the plant cell might be making glucose in photosynthesis. They need to store it to stop osmosis happening, and obviously we've got a chloroplast which does photosynthesis. And I've just made a video, the introductory video of photosynthesis for year 13 if you want to know more about chloroplast. But you can see an actual chloroplast here. So chloroplasts are huge structures in plant cells. So that's a chloroplast there. There's another chloroplast down here. Okay, so a lot of people, sometimes people underestimate the size of the chloroplast, but they're absolutely huge molecules, huge parts of the cell, huge organelles, sorry. Okay, now sort of focus organelles which you need to make sure you learn about, which we've already spoken about, are these ones. So you've got the nucleus, obviously, which often has rough endoplasmic reticulum lining around the outside, and that's because we've got messenger RNA takes the information from the DNA of the nucleus to the ribosomes to make proteins. So it's handy there on the outside. You've then got the mitochondria, which is involved in respiration. Again, much more important to learn its structure for year 13, but you could be asked about its structure in year 12 as well. The chloroplast, which is involved, again, a lot of year 13 content in photosynthesis. And then you've got the Golgi apparatus, which is just membranes, are involved in lipid packaging and processing proteins. So just a summary of some of the roles then. So smooth endoplasmic reticulum is a system of membranes. It doesn't have ribosomes attached, and they make steroids and hormones or fats, basically. Rough endoplasmic reticulum has ribosomes attached to the membranes, and they make proteins. Golgi apparatus is a load of membranes that manufacture or modify proteins and package them, and might add lipids to them to make something called glycolipids. Mitochondria carries out stages of respiration. Ribosomes are protein synthesis. Lysosomes contain these hydrolytic enzymes that can break down bacteria, but in sperm, as an example, you have an acrosome which breaks down the egg outer layer, so that shows they can digest. Chloroplasts obviously contain chlorophyll, do photosynthesis. The plasma membrane, or the cell surface membrane, controls what enters and leaves the cell, and the nucleus is obviously the control center of the cell that contains the DNA, which helps us, or makes the codes for our proteins. You've got cilia, which are short extensions of some cells, and it can move fluids over the surface of the cell, or it can help move the cell itself if it lives in fluid. You've got flagella, which are long thin extensions and can again cause the cell to move. You've got the nucleolus, which is the darker part of the cell, and it's used to make ribosomes. You've got the nuclear envelope, which controls what enters and leaves the nucleus. You've got the nuclear pores, which are holes in the nuclear envelopes, which allow small molecules to enter and leave the nucleus. And you've got the vacuole, which controls osmotic potential in the cell, controls osmosis in the cell. So you might get a question that looks like this. It says, describe the division of labour involved in the manufacturing and the release of insulin. 7-mark question. Now, if you've got this question, it would want you to talk your way through it. Now, it might not be a question you can necessarily do very well at the minute, but you'll understand it much more later in the year. But based on what we've just gone over, you should be able to sort of link some answers together. So if we start off then, what we've got is, first of all, the question is asking you about insulin. So insulin is a hormone, and most hormones are proteins. So the question might say proteins directly, it might say hormone, it might say insulin. So because it's a protein, then it's going to have a particular way of being made. So the first thing you need to say is that one, and you will learn some of this later on, is that one gene of DNA, which makes the protein insulin in this case, unwinds in the nucleus. And that makes a molecule of something called messenger RNA. Now, you don't need to know exactly what that is to start off with, but you will do later on in the course. Now, that messenger RNA, which carries the code of one gene, which is the code to make one protein of insulin in this case, is small enough to fit through the nuclear pores and moves to the ribosomes that are on the rough endoplasmic reticulum. They make the protein insulin, and then the protein insulin can be released into the bloodstream from this would be something called the Langerhans cells in your pancreas. However, you can't just release them straight into the bloodstream. So they have to then go to the Golgi apparatus where they are packaged to make sure they don't just dissolve in the blood, okay, and they are modified. So Golgi apparatus takes that protein, packages it, and modifies it, and to help it leave the cell, it wraps it in something called a layer of fat, and we call that a vesicle. And the vesicle then leaves the cell from the Golgi apparatus, and it moves and it merges with the cell membrane, doing something called exocytosis. Now, this cell, part A of the organelle here is the mitochondria, so that would use lots of energy to do that process. And you might get a sort of example like this, which sort of talks you through it. So if we start off here, this is like a highly detailed one that you hopefully be able to do later on in the course. So messenger RNA copies the gene from the nucleus. That messenger RNA then leaves the nucleus and it goes to the rough endoplasmic reticulum, where it then makes a protein molecule. That then goes to the Golgi apparatus, which then modifies and packages the protein, and then that leaves via a vesicle, and it goes via the external membrane, so which is called the cell surface membrane, via exocytosis, releasing it into the bloodstream. So what do you need to know to take away? You need to know what the functions of the organelles are, and you need to know how the organelles work together in this thing called division of labour. And this is a typical example on the screen now, where you'd be asked, how is a hormone or a protein made in the cell using division of labour? And you'd be expected to describe what the nucleus does. That then goes to the rough endoplasmic reticulum, which makes proteins. Then what happens at the Golgi apparatus, and then about the vesicles and exocytosis. So I hope that's been useful for you guys. I'll see you again soon.